evening, or this afternoon. Dr. Lynn Adler, who's a professor of biology at the University of Massachusetts Amherst. On screen here. Her lab researches the ecology and evolution of plant-insect interactions. She has fascinating research, a large lab with lots of graduate students and postdocs that have gone through there. Many successful students who have gone on to sort of carry on this type of research at other schools around the country. Lynn did her undergraduate work at Brown University, and then did her PhD at the University of California in Davis, one of the top ecology schools in the country. Dr. Carbon?
Rick Carbon and Sharon Strauss.
Okay, Rick Carbon and Sharon Strauss, two also very fascinating ecologists. And then she did a postdoc at the University of Arizona before coming to UMass in the entomology, plant science, plant, soil, insect department before it was merged with the biology department recently.
Okay.
All right. Well, thank you for coming out.
Thank you. Thank you all for having me. It's a pleasure to be here and a real pleasure to get to talk to this audience. Will it be okay if I'm over here? So you're going to focus on that, right? It's okay that I'm standing here? And everybody can hear me okay? No problem in the back? Great. Awesome. So I like to start with this absolutely ridiculous picture that shows on my PowerPoint skills, a kind of a way of thinking about the world a little bit. So the vast majority of multi-celled species on this planet are plants, but also, insects. So plants and insects together make up about 75% of the multicellular species on this planet. So interactions between plants and insects take up a lot of ground, take up a lot of space, right? And way back in the dark ages, when I was first starting graduate school, people tended to kind of divide themselves up. So there were all of the people who thought about insects that eat plants, and all of the ways that plants have to deal with that. They mostly thought about this in the context of leaves. So we would think about the defenses, the way that plants try to keep insects from eating them. Mechanical defenses, but also chemical defenses. We would think about how those defenses would evolve in response to pressure from herbivores, and we would think about how herbivores would evolve in response to pressure from these defenses in plants. And that was kind of one part of the world. And then there were all the people who think about pollinators, and think about flowers, and think about the shape, and the size, and the color, and the rewards, and maybe the scent, and think about how pollinators are going to make their foraging choices in response to all of those signals and cues from plants, and how those floral cues in return are going to evolve in response to the decisions that pollinators are making. And way back when I started grad school, these were like two separate worlds that didn't really talk to each other very much. And one of my PhD advisors, Sharon Strauss, was one of the first people to kind of say, hey, wait a minute. This is all happening through a shared plant, and there's got to be ways that these different spheres are going to interact. And since then, she and I and many, many other people have thought about some of the different ways that there's actually much more integration here than you can think about. So for starters, damage to a plant is going to affect floral traits. Right? It's going to change resources available for those flowers. It's going to change shape, size, rewards, all of those things we now know damage to a plant can affect. In addition, the more we look, the more we find that those chemical defenses we've studied for so long in the context of leaves and leaf damage are present in flowers, are present in nectar, are present in pollen, and so have the potential to influence interactions with pollinators and maybe even evolve in response to interactions with pollinators, as well as influencing interactions with herbivores. And then we could also imagine that herbivores might select on the evolution of defense in plants. If pollinators don't like to visit damaged plants, that may be a selective advantage for plants that are well defended. Conversely, if pollinators don't like those defenses, that may be a disadvantage for those plants. And then, this isn't a good example of it, but I've got right here, but sometimes herbivores and pollinators aren't separate organisms at all, right? You can imagine that this caterpillar is going to, if it's lucky, at some point become a moth that is also a pollinator. And so from a plant point of view, there may be a challenge if I need to try to keep away the caterpillar, but attract the adult. So not separate plants and herbivores. And then, of course, course, the traits we study in leaves and the traits we study in flowers are not separate and independent of each other. Resource allocation, pleiotropy can all link the expression of traits in different tissues. And then let's not forget that my ridiculous PowerPoint plant doesn't even have roots. And there's a whole other sphere of interactions that's happening between what's going on above ground and what's going on below ground. So this is the kind of stuff that I've been doing that keeps me out of trouble. All those different links I've made. I've done some research on each of those parts over the years, but in recent years, I've come to recognize that even this sort of big picture is not big enough. Because all of those interactions are embedded in a larger food web, right? And the people who study herbivores know a ton about this. They've been doing this forever. So we know about predators that eat herbivores. We know about parasites, parasitoids that attack herbivores. We know how plants can affect these interactions. We've been studying that for a long time. But pollinators are embedded in their own food web too. They have predators like crab spiders, they have parasitoids, they have pathogens that may also be influenced by these tritrophic interactions. And that's what I've been focusing my energy on for recent years, is this sort of interaction between flowers and pollinators, and particularly a pathogen that commonly interacts with these pollinators. So just to kind of reiterate this a little bit more, yeah, the herbivore people have known for decades that plant defenses mediate tritrophic interaction. By the way, do you know what this is? You guys are botanists. It's a beta hormone, awesome.
We actually saw this today in one field.
Did you guys?
Yeah.
Yes, cool. What is it then? Yeah, it's cocoons, right? Everybody says eggs, but they're these wasp cocoons, right? So the wasp lays the eggs inside the caterpillar, they hatch, they eat the caterpillar out. When they're ready to pupate, they come out, make these cocoons. So this is a tobacco hormone, actually, and the nicotine from the plants can mediate that interaction that the caterpillar is having with the wasps. So we know that there's lots and lots and lots of examples. It's kind of remarkable, in fact, given how difficult it is to come up with generality in ecology at all, for how many different systems we know that defenses, generally directly reduce herbivore performance. That's kind of what they're there for. But they can indirectly benefit herbivores by protecting them against their predators, parasites, and diseases. So many different plants, so many different chemicals, so many different herbivores. We kind of see that this is the case. And that's kind of cool because, these days everybody's concerned about pollinators and save the bees and all of these issues. And there's a lot of stresses that the pollinators are dealing with. But let's also remember that they're kind of an herbivore, right? Like, from the plant's point of view, the interaction with the pollinator is different from the interaction with an herbivore. But from the insect point of view, this is a really specialized herbivore that just eats nectar and pollen, which are plant tissues. And the more we look, the more we find that nectar and pollen contain those plant defenses we've studied for so long in the context of herbivory. And although there's a lot of things involved in pollinator decline, parasites and pathogens are on the list as some of the stresses that are influencing pollinator populations. So we wondered, can we take what we already know from plant herbivore natural enemy theory, literature, and apply it to understand something more about plant pollinator parasite dynamics? And when I say we, it means it's not just me. I have collaborators, Becky Irwin at NC State, Jay Evans, who's a honeybee biologist at USDA, Phil Stevenson, who's a biochemist, who are all collaborators on this project. So to kind of take all that hand-waving and turn it into some specific questions, the structure of the talk today will be, first of all, yeah, it's a nice idea, but I have great ideas all the time that turn out to have no foundation in reality. So do we find that consuming nectar secondary compounds actually does have an impact on pathogen loads in bees? And then moving from there away from nectar, oh, excuse me, trigger finger, towards pollen. What about pollen consumption? Does that affect pathogen loads? And then kind of broadening out a little bit away from this focus on nectar and pollen chemistry to think more generally about the role that plant species might play in their ability to transmit pathogens between floral visitors. And then finally, if plants do matter in those transmission networks, does that variation in transmission actually scale up to affect colony level diseases and bee performance? And the cool thing about this last question is we can kind of take the same question and frame it in different ways for different audiences. So like an ecologist might say, okay, this question is kind of about, does community context matter? Do the plants that you have in your community have an impact on bee disease? But if we're really interested in applications, we can kind of think about that question in a different way, which is, can we help manage pathogens in bees by what we choose to plant in our pollinator habitats? So just to introduce you a little bit to the players in this system, the vast majority of work I do is on the common eastern bumblebee, Bombus impatiens. It really is very common throughout the eastern U.S. It's a great species for us not only because it's so prevalent, but also it's commercially reared, so we can get colonies in the winter. Anytime we want them, they come to us in boxes. And I like to acknowledge that this species is not in decline. This species is doing great. So while we use this species as a model for understanding how plants might mediate interactions between pollinators and pathogens, we are not trying to understand why this species is in decline, because it's not. The pathogen I've been working with is, doesn't have a common name, sorry. I'm going to call it Cryptidia. It's Cryptidia bombi. It's a trypanosomatic gut parasite. It's related to parasites in humans that cause Chagas disease, leishmaniasis, African sleeping sickness. It's transmitted when an infected bee defecates, there's infective cells in the feces. And if another bee consumes that, then they can get infected as well. So it can be transmitted both within the hive, but potentially outside the hive. It's not the baddest of the bad as pathogens go. It's not a death sentence if you're infected with this pathogen, but it does decrease lifespan, it decrease colony reproduction, decrease the odds that a queen is going to make it through the winter. It's not good, but it's not the worst of the pathogens out there. But it is extremely common, particularly in this area for some reason. We can find it in up to 80% of, that should say bumblebees. It infects bumblebees in western Massachusetts in this area. So now that I've kind of oriented you to this system, when we started thinking about these questions, the very first thing we wanted to do was look at nectar secondary compounds and how they might affect pathogen loads in bees. So we did a really pretty straightforward lab experiment. First, we combed the literature to find as many records as we could of chemical defenses in nectar. There's really not that much out there that actually reports the concentration and that were compounds that we could obtain commercially. We came up with 8 separate compounds that we could work with that we tested in separate experiments. And the experiments are really pretty straightforward. We take bumblebees and we put them in separate little individual vials, and we give them a nectar feeder, which is just a little microcentrifuge tube on the end there. And we have lots of them. And we start out by inoculating everybody with a dose of Cryptidia. So everybody gets exposed to the same amount of pathogen to start with. If we want to figure out what's going to make them better, we have to start by making them sick. But we start everybody off with the same dose. And then we randomly assign them to treatments. It's a good classical experimental design. So they might get just a sucrose solution, or they might get a sucrose solution with one of those 8 compounds at whatever the relevant concentration was that had been documented in those species. They get fed that diet for 7 days, and then we dissect them, and we look at the pathogen load of the guts. I don't know whether you guys care or not, but these are the compounds that we tested. They're fairly broadly distributed across plants and across chemical structures. Some alkaloids from citrus and tobacco, cyanogenic glycosides from almond, iridite glycosides that are in a lot of plants, but we took our concentrations from turtlehead. There's tons of phenolics in buckwheat and lots of other things, and thymol, which is actually used as a mite treatment for honeybees, that is in thyme flowers and in basswood nectar. And this paper came out a few years ago. We did these 8 separate experiments. We did some of them at Dartmouth, where my collaborator was at the time, some at UMass. We replicated some of them, and then this is one of those painful things where it's like one figure that summarizes all of this work. And these are the compounds. And the way to read this is pathogen loads. We did a meta-analysis because we did these all in separate experiments. And so what you're seeing here is a comparison of the treatment effect against the control effect. If the dot is under the 0 line, that means the treatment suppressed the pathogen relative to the control. Okay? And what you can see right off here is that 4 of these, they're all below 0. So every single one, the treatment had lower pathogen loads than the control. These 4 confidence intervals don't overlap 0. So those are significantly so. So 4 of these 8 compounds significantly reduce pathogen loads, cell counts, relative to control treatments. And you can't really tell so well from these effect sizes, but this is about like a 60 to 80% lower counts in the bees. So this was exciting. We did this first experiment. We found, wow, these chemical defenses that bees are encountering just from foraging can actually play a role in reducing their gut pathogens. 4 out of the 8 significantly reduced pathogen loads, ranging from 60 to 80% compared to controls. That suggested that nectar chemistry might play a really important role in mediating bee interactions with their pathogens. We were so excited. We wrote, we got an NSF grant, we got a USDA grant. This is going to be great. Okay, now let's go get started. And then sometimes when we went and did the next step, sometimes we'd find that those chemicals had effects, and sometimes they didn't, and we'd scratch our heads. And maybe we got really, really lucky with our Chlamydia strain the first time around. It seems like strains actually vary quite a lot in their sensitivity to these nectar compounds. And sometimes we'd see the effect, sometimes we wouldn't. And we said, okay, shoot. So maybe we're missing the boat here. You know, pollen is often a lot more chemically defended than nectar is. Kind of makes sense, right? That's the gametes. I wonder if there's anything interesting going on with pollen. So we did the same kind of experiment as that nectar experiment. There's not that many choices out there of monofloral pollens you could find. There's a pretty limited list. We picked a few off of it. We got sunflower pollen, buckwheat pollen, canola pollen, or we had a mix of all 3 in equal weights. These were each species. We thought there might be something interesting about them. Just for fun, this is what it looks like. Sunflower pollen is so nice, bright orange. Here's a whole bunch of bees in the experiment. And this was an undergrad honors project in my lab. Jonathan Giacomini did this for his undergrad honors thesis, then he went on to grad school with my collaborator. And after a while, I was like, hey, Jonathan, how's it going? How's it look? He said, it's so weird. So this is really weird. But when the bees are fed sunflower, they just don't have Chlamydia anymore. Whoa. So this is a graph that's easier to read. Chlamydia count, that's per 0.02 microliters. And this is the number of cells. So buckwheat, canola, whoa, sunflower. So much more dramatic than anything we've ever seen in nectar. And the mix is low, but not as low as just having pure sunflower. And what's more, it's just a lot of bees we didn't see an infection in them after a week. So this is the percent of bees without Chlamydia, and after a week, they were all infected to start. But after a week, 2/3 of the bees fed sunflower, we just don't see the pathogen anymore. The ones that did have it, it was very low. About 10% each of buckwheat canola, and about 1/3 in the mix. And so that result, I'm happy to report, has been very consistent. We have done this many times with sunflower from many places. And yeah, sunflower is kind of magic. So we started to wonder, did we just get lucky with this one cultivar of sunflower that we happened to purchase, or is this something that's a little bit broader? So sunflower is amazing. There's hundreds and hundreds of different domesticated cultivars, and then of course there's wild. Sunflower is native to North America. There's wild populations, and then lots of congeners. So a master's student with me, George Lacasio, took on the crazy project of growing lots and lots and lots of different cultivars and species. And the thing is, people say like, oh, test this pollen, test that pollen. It's not easy to get enough pollen to do bioassays with bumblebees. Like, that is not a small job. We had the brilliant idea that we would grow the plants in tents and put honeybees in them and get them to collect the pollen for us. And it turns out honeybees don't like tents. They need really big tents, like wedding gazebos, and even then they're kind of cranky about it. That's George celebrating the most expensive pollen you have ever seen collected. We did a lot of hand collecting. This was yet another of Lynn's brilliant ideas. So this is another graph that represents a an unbelievable amount of work. But here's our Cryptidia count on the y-axis again. This is buckwheat. You remember buckwheat was the one that had the highest counts before? Maybe you do, maybe you don't. Buckwheat had the highest counts. This is a wildflower mix, so just wildflower pollen you can buy. And here's everybody else. 17, if I recall correctly, different species, cultivars, they all reduce Cryptidia relative to buckwheat and wildflower, all of them. Some slightly more than others, but all of them do this. Even, we had an opportunity to collect some goldenrod pollen, which is in the same family as sunflower, but not that close. Goldenrod pollen did it too. We haven't gotten a chance to collect from other composites that are in the market. If you know a big field of black-eyed Susans, echinacea, anything else, I want to go put some honeybees on it and see if we can get the pollen. So it seems like this is something that is both repeatable and perhaps widespread in composites. And that's nice, but it was all done in the lab. This is actually matter out in the field. So we wondered about whether we could actually see this signal on more of a landscape scale. So we sampled bumblebee workers from about 20 different farms and then we measured how much sunflower was growing in each of those farms. So this was kind of a last minute thing. As you can imagine, there's going to be tons of noise because we didn't control anything else about what was at those farms, how big they were, anything about where the bees came from. We just went, grabbed bees, took them back, counted their Cryptidia. So the data are understandably messy, but the more sunflower at the farm, the lower the counts of infection, which suggests that something we saw in the lab may actually play out at a larger scale as well. So does pollen consumption affect pathogen loads? Wow. Consuming sunflower pollen dramatically reduced Cryptidia infection in individuals. Oh, I'm sorry, I took out some data, but also we made little miniature microcolonies of these, reduced it in those as well. We found that multiple types of sunflower were effective. We found that more sunflower on farms meant lower counts of this pathogen. Based on those initial results, we got a pollinator health grant from the USDA and have ongoing work now, first of all, developing applications. So can we figure out how much sunflower we need at a farm to be effective? The exciting thing about sunflower is that because it's both a crop and a native plant, I think it has the potential to be incorporated both into agroecosystems and into native pollinator habitat in places where that's appropriate. We're also looking at developing sunflower supplements as a non-chemical way for bumblebee commercial companies to be able to manage disease, which they seem very interested in. I would like to see how far this goes. I would love to work with more composites, more bees, more pathogens, ideally. We've stumbled across this in this one species interaction, but how widespread is this effect? We have some suggestions that it may be much broader than just the species. And then, of course, everybody always says, what the heck is it about sunflower pollen that makes it such magic? And the answer is, we don't know. A couple honor species later, I can say it's not this, and it's not this, but we're still trying to figure out exactly what it is. We have a new USDA grant, luckily, to allow us to sort of focus more specifically on elucidating the mechanisms. So that's a lot about nectar and pollen. And now I kind of like to step back and think a little bit more broadly about the role that plants can play in how diseases get transmitted. And this is part of a grant that, of all things, is funded by the NIH with Scott McCart at Cornell University as the lead, Quinn McFrederick, Steve Adler, Chris Myers, Peter Greystock, and others. And so now, you know that moment, like you're in the airport, or hey, maybe you're at a seminar. Somebody just said like, oh, like a disgusting public bus. Yeah, like you're in a crowd of people. And then all of a sudden the person next to you, and you go, ugh. I'm gonna get sick, and I'm gonna take it home, and I'm gonna infect my family or my friends or my roommates, and this is gonna be miserable for everyone. Right? So places that have high densities of organisms, all mixing and sharing their germs and then dispersing are really interesting models of disease transmission that are relevant to many, many systems, including humans. And then you think about flowers, which can have 10,000 times the density of insects as the surrounding foliage. Places where insects are coming in, they're feeding, they're pooping, they're dispersing back to other places, they're moving to other flowers. So flowers might be able to serve as hotspots of disease transmission to floral visitors is an idea. There's not a heck of a lot of data out there to actually support it, but some increasing studies, a seminal paper that came out 25 years ago did show that, that this very gut parasite, Crithidia, can be transmitted between infected individuals. If you let an infected bumblebee forage on a flower, and then you let a healthy bumblebee forage on it, there's a decent chance that that healthy bee is going to get infected too. 25 years ago, and the rate was different on 2 different plant species. Cool. And then like almost nothing for decades until people start to get concerned about pollinator decline. And so now there's starting to be some more studies. A cool one in 2010, 2015, a really nice study, but basically showing that bumblebees and honeybees can vector and transmit diseases between them as well, potentially using flowers as the site. But still, for the most part, if you buy a bumblebee colony, like these the ones we get from BioBest, and it's healthy, and you stick it out in the field, sooner or later, these are going to start bringing back infection to the colony. And we really know almost nothing about what it is, what characteristics of the landscape might affect how much infection is getting brought back to that colony. So to try to think a little bit more about the role that plant species might play, we started by just picking 15 different plant species that we grew in a common plot. We mostly picked them off of those like pollinator-friendly planting lists, what to plant to support your pollinators, but we also chose some cherry-picked some invasives and some horticultural species we were interested in. And we would put bags over the flowers to keep wild bees from visiting. So we wanted to prevent any wild bees from depositing pathogens onto the flowers. And yep, this came out last year. Finally loaded up. This is Sarah Junkanen, who was an undergrad who helped with this, and a gorgeous pollinator garden. And to conduct what we call a transmission trial, we would take one of those inflorescences, and then we would inoculate it. So we would put 4 drops of Crithidium inoculum on that inflorescence in flowers, so that everybody's starting out with the same, ideally, potential to infect a bee. And then we would allow one healthy experimental bee to forage on it. We made sure it consumed at least one of those drops of inoculum. And then we take it back to the lab, rear it for 7 days, see whether it got infected or not, and how bad the infection was. Okay? And then we could ask, okay, do plant species differ? Is the odds of getting infected different if you forage on a different plant species? This is kind of what a trial looks like. This was a high school student, Twin Chuang, who was helping that summer. This is a cute bee foraging on a penstemon. He's just, I don't know, it never grows old. And then here's a bunch of data. So, if we just look at sort of overall transmission, these are sort of 3-letter codes for each of the species. Milkweeds up here, the callus way down here, and here's the mean count of pathogens, including those that just didn't get infected at all. What you can see is that there's like a 4-fold difference in how much you get infected depending on who you were foraging on. I'm happy if you guys are botanists. You want to know which species these are. I can tell you at the end. And if you kind of squint, you could almost say it looks like there's a bunch of species that really don't transmit very much. Oh, I know I was going to lead you guys here. And then there's a bunch of species that seem like they really transmit quite a lot. Okay, I want you to remember that. Low transmission and high transmission plant species, because that's going to become important. It's very well good that we did this for 15 plant species, but it's not that useful. We can't extrapolate beyond that. So we really wanted to know whether particular floral traits would predict transmission. So we measured the length and the width of the flowers and nectar production and shape and all of the chemicals in the nectar. We did all this to measure floral traits, size, number of open flowers, nectar volume, height, chemistry, and yeah, and I never know how to, like, we got a very weird result. Traits overall explain transmission better than species do, but none of the traits I thought might matter seem to matter. The only thing that seems to matter is how many reproductive structures per stalk plant has. More and like that's like buds, flowers, fruits, everything. Not the number of open flowers on the one that the bee was actually foraging on, but like total reproductive structures. More of those, more transmission. I'm baffled by this, and I'm happy to hear thoughts. Yeah. So do plant species vary in their ability to transmit pathogens? They do. We found about 4-fold variation in transmission and quickly across plant species, even though we controlled how much inoculum was on those inflorescences. So everybody had the same amount of inoculum, but the odds that the bee would get infected were quite different. This suggests that plant species identity could modify bee pathogen dynamics, maybe reducing or, hey, increasing infection in bees. So that leads us to our last question. Could that actually be going on? And I think that this matters particularly. This is not a great picture. I apologize. I took it. This is a cornfield in upstate New York with a row of sunflowers along the edge. So people are investing a lot of money and energy and resources in pollinator habitat. And I think that's a good thing overall. But if some species might play more of a role in disease transmission than others, it seems like that might be worth knowing. So the design of this was to assess whether our choice of, I'm going to call it hedgerow planting, those kind of pollinator plantings that you put up, could exacerbate or ameliorate the pathogen loads. This was a USDA-funded project. We used canola as like a focal crop. It's self-incompatible. We were interested in pollination services. None of that came out very interesting. And we did this experiment in tents. So we set up tents as our unit of replication. Each tent had one bee microcolony and a bunch of flowering plants. This is nuts. This was truly crazy. We spent the summer doing this experiment going, this is crazy. I can't believe the USDA actually funded this. Like, this is nuts. It really was. So, okay, and this is the one piece here that isn't published yet. So just to tell you what microcolonies are. One of the many cool things about bumblebees is, so it's hard, you're not going to get enough food to feed a whole bumblebee colony into a tent. Like, that's just not going to happen. It takes too much food, unless it's a ridiculously humongous tent. But you can make little miniature colonies. So you can make what we call microcolonies that we put maybe 15 workers into a deli cup. And one of those workers will start to act like a queen if there's no queen in the colony, and she'll start laying eggs, and the others will do foraging and do brood care. Because bees are haplodiploid, that worker hasn't mated, so she'll only lay males. But this is a way to sort of get a little miniature version of being able to ask questions about what might affect bee reproduction, as well as just looking at pathogen loads. So we made little microcolonies, and we would put them in cardboard boxes to stick them out in the field, and this is an undergrad, Yunjin Moon, modeling our wonderful field-ready boxes of microcolonies. And now to think a little bit more about the experimental design, we had 3 treatments. So some of these tents just had canola, nothing else, to mimic a farmer that is just planting canola. And then we had 2 different versions of what I'm going to call our hedgerow treatments. So we had one that was primarily low transmission plant species. Remember those low and high transmission plants? And I said, remember this? This is why it's important. So we picked species off those lists of low transmission or high transmission. They were chosen based on a prior experiment. We did multiple rounds of this experiment. It was crazy. And depending on what was flowering at the time, we picked different species, but always either from the low transmission or the high transmission. We also manipulated whether the bees in each tent were infected or not. So half the colonies, microcolonies, were infected to begin with, and the other half weren't, because we were also interested in whether infection influenced pollination service and behavior. I'm not going to be reporting on those results today. So again, tents are our unit of replication. So we have 6 combinations, right? 3 hedgerow treatments, infected or not, 6 combination. 3 replicates of that per round, so 18 tents in a round, and we would set it up, run it for 2 weeks, and then take it down, dissect all the bees, and do it again. We did it 5 times. over the course. Yes, this was crazy. This was really nuts. This experiment, I almost drove me nuts. Just to visualize what these treatments look like, if you were looking down on the tent, a canola tent would have a bee microcolony in it, and then it would have 15 bins of canola, and that's it. A low transmission tent would have a bee microcolony in it, 12 bins of canola to account for that if you're allocating part of your field to a hedgerow, you have a little bit less to plant in your crop. And then 12 plants here, and I'm using each symbol as a different species, and the color indicates low or high transmission. So now I have 1 high transmission species and then 3 low. Okay? 1 high that's different, another 3 of these same low. 1 high of the 3rd species and another 3 of those low. So we have 3 high transmission species and 3 low transmission species, but we've got 3 times as many of the low transmission as the high. Whereas in a high transmission tent, we've got the bee microcolony, we've got the canola, and then we have those same species, but in a reversed ratio. So we have the same 6 species in these tents, but we have a shifted ratio of high to low transmission species. Does that make sense? Awesome. Okay. So yeah, low and high transmission, same species, but reserve reverse ratios, 3 to 1 or 1 to 3. This is what a bunch of tents in a field look like. That's a bunch of bins of canola in a tent, and here's our bee microcolonies set up on a little stick off the ground to keep ants out, and some gorgeous hedgerow plants. Great. And then, yep, once again, nice simple graph from an insane amount of work for the summer, but oh my goodness, this worked. So here's our Crithidia count again, the cells per 0.02 microliter. This is averaged over all the rounds. So all those different high and low transmission species, this is the summary for everything. If you put high transmission plants predominantly in the tent, you have about twice as high of a pathogen infection in those bees at the end of 2 weeks than if you put low transmission species in the tent. And canola is somewhere in the middle. So I'm so excited about this. So excited. So using plants from our silly little transmission trial assays that increased transmission actually increases transmission at an entire microcolony over a 2-week period of foraging. And furthermore, this isn't a diversity effect, right? Like if diversity were what mattered, you would think that both the hedgerows would have lower infection than the canola. But that's not what's going on here. High transmission plants have the highest pathogen load, even though they're more diverse than just having canola in the field. Unsurprisingly, hedgerow treatments also affect the performance, so we can look at the reproduction in those microcolonies over the 2-week period. We looked at lots of different things. So here's the survival of the adult bees. They don't do as well when there's only canola in the tent as compared to high or low, and there's no difference in high or low transmission. Even though those high transmission tents have more infection, it wasn't scaling up to affect the survival of the workers. They actually do great on those high transmission. This is larvae produced. Yeah, they don't do particularly well on just canola. I will acknowledge that these are bees that were constrained to only forage in these tents. They were probably food limited relative to what they would be in a more natural situation. So, yeah, that seems like a likely scenario. So yeah, we thought, what's going on here? Why do they do so well on these hedgerows? Maybe it's the amount of food that they have. And indeed, because we did a lot of pollinator observations and we counted how many flowers of each species were open each tent every time we did observations. And we also knew what the average nectar standing crops were for the flowers of each of those species. So we could calculate how much nectar was available in those tents several times over the course of each round for each tent. And yeah, canola's terrible. Like, it just doesn't have a lot to offer these bees relative to these hedgerows. And in fact, we find that if we add nectar to our models, so if we both look at treatment and nectar resources in our models of performance, bees do better when they have more nectar. Here's that adult bees, and here's that larval produced, and each of these colors represents a tent, low transmission, high transmission, canola only. And if we add nectar to the model, the effect of treatment is no longer significant, of hedgerow treatment is no longer significant. So nectar, flower, food resources are what's explaining the effect of hedgerows on bee performance, on adults, on larvae, on pretty much anything we look at, but not infection. The amount of nectar in the tent did not predict the infection rate in that tent. So something different is going on about what influences reproduction in those bees and what influences transmission infection in those bees. There are different mechanisms happening here. So does variation in transmission affect colony pathogen levels and bee performance? Disease transmission ability in hedgerow species, to our great astonishment, accurately predicted colony level disease loads after 2 weeks of exposure. But I'd also like to point out, bees did best when they had hedgerows. Even if they got more infected, they still did better having those pollinator resources than not. So resources, oh, excuse me. Is that coming up? Okay, excuse me. Resources explain the effect of hedgerows on bee performance, but not pathogen infection. So more resources, being bees that survive better, reproduce more, but there's something else about those plants that shapes how much those bees either transmit or maintain their infections. So to conclude, I'd say that nectar chemistry and pollen can both strongly affect bee pathogen loads. So there's a lot of people who are looking at things that might be important for bees, but sometimes variation in plants is left out in that picture. But in fact, it looks like resources that bees are getting from different plants can have a big impact on their disease loads. Pollinator-friendly bee plant species differ widely in their ability to transmit pathogens between bees, which is potentially maybe mediated. I mean, it's got to be mediated by floral traits at some level, but we haven't yet figured out how. Co-flowering plants improve bee performance, so the choice of hedgerow or not hedgerow, it's better to have those species, but some species also seem to increase the pathogen loads. So the choice of species can affect disease loads, potentially in this sort of mesocosm agroecosystem. And so the take-home I'd like to leave you with is this idea that floral traits and plant community composition may play a large and often largely unrecognized role in mediating bee pathogen dynamics. And with that, I say thank you, and then I'm also going to ask you to indulge me. I'm one of my midlife crises I've been trying to use other parts of my brain, and so I invite all of you to join the fun. It's a real challenge to try to sometimes translate our science for the public, and especially to condense it into really short formats. I find that some people are haiku people, some people kind of are a little more like limericks, are their speed, but I have a science poetry Twitter account now. So you can take your previous or your next publications and see how you can do in condensing it down to a 575 syllables. It's fun. Once you get into it, you can tweet it. If you're old school, frankly like me, you can email it to me. You can check it out on my website and see what other people have done. Have fun. I do. And with that, thank you to many funding agencies, to collaborators, to many student researchers, grad students in bold, undergraduate co-published with Asterisk. Thank all of you for your attention on a Saturday afternoon, and I'm happy to take questions.
Recite us a haiku or a poem, please.
Recite us a haiku or a poem? Right now, off the top of my head. Oh my goodness. Check it out. Can I like look what, it's been like a couple weeks since I've wrote one, and I can't even tell you what I ate for breakfast this morning. But just hold it right to the end. Okay, let me get the other questions. I'll do it for you. I got some. I'll put it up. So it sounds like I need that on the next talk. I need to put some of them up there. It's fun at like conferences. People start, if I go early, then people start actually writing their own and putting it at the end of their talks. It is fun. But yeah, I'll get some. Okay, sir.
Were the invasive plants that you used, they fall in either the high transmission or low transmission?
We have our blue strife, and that was pretty high transmission, lots and lots of flowers. I'm trying to remember if we had any, but we had Linaria vulgaris, and that was pretty low. It's got some pretty nasty chemicals in the nectar, and it's kind of low. So it wasn't like invasives as a group had any particular pattern. No. Yes.
For the types of like high versus low transmission, I think you had sunflowers in there.
Mm-hmm.
Where was that exactly?
No.
It was very low, or was it like, it's not like the lowest though?
No, it wasn't the lowest. There
I saw
was a whole bunch that were kind of low and not that distinguishable from each other.
Okay. Sorry, I'm making you go all the way.
No, it's cool. I'm gonna have to look at the poetry. And you guys are a captive audience. I've got nothing else to do. Ooh, you got one. You're so great. Thank you. Yes, there. Sunflower. I mean, not
The lowest, but it's still
particularly,
pretty.
and
With the
statistically
bars,
no
yeah.
different from any of the rest of those. There's a lot of variation in cryptidae accounts that would get too hung up on very small details. Yeah.
Do you know if there's any difference in pollen composition between plants with a lower number of reproductive structures and a higher number of reproductive structures?
Oh, that's a good one. I went down a bit of this like rabbit hole on this thing because it was making me crazy. I was like, what would be so different? And I decided maybe if you have lots of flowers, they don't last very long. And maybe if you have a few flowers, they're open for longer. And okay, so humor me on this. And if they're open for longer, that you'd have more need to defend against microbes, right? Because like a flower that's sitting open for days. So like maybe the flowers that are open for a long time are more antimicrobial. So then I kind of measured on the species where I could still get hold of it, how long are the flowers open, and that didn't predict anything either. It's not really answering your question.
Well, is it genetic? Do genetics determine the number of reproductive structures or have an influence on that?
This was sort of in a cross-species comparison, so I would think so. I mean, but at a pretty large scale.
I was wondering if there might be a genetic linkage between whether it produces, how much of this compound it produces, and the number of structures.
But which compound?
Yeah.
Yeah, nectar chemistry turned out to be interesting, but there are hundreds and hundreds of compounds, and they were, I think the most that any of these species shared compounds was maybe 2 or 3 would have the same compound. So when we tried to really look at nectar chemistry, it was very hard to do it with only 15 species. 2 or 3 would have a particular alkaloid, 2 or 3 would have, there's a bazillion different phenolics, even categories. It turned out to be a real snarl to try to disentangle those roles. Yeah. Yes?
I was going to say, while you're on this slide, would you go ahead and identify,
my goodness.
just to be sure everybody
Sure.
knows?
So Digitalis purpurea, foxglove, Antirrhinum majus, snapdragons, Linaria vulgaris, buttered eggs, Thymus vulgaris, so thyme plants, Patience capensis, jewelweed, Helianthus annuus, sunflower, Eupatorium, oh my goodness, what's this? Joe Pye. Thank you, Joe Pye, that one. This was goldenrod, which is interesting because that was high transmission, even though the pollen we find by itself suppresses. Penstemon, Digitalis, Proboscis, Lobelia, the blue one, Syphilica, Lythrum, the purple estrife. This was bee-balm, Leonarda, and this was swamp milkweed, Asclepias incarnata. Yeah, sorry, I knew I was going to have to do that, right?
For botanists, you got to.
Yeah, I'm happy to. I'm doing insect stuff these days, but I think my first love is still the plants. Yeah.
So I was thinking about the low transmission pollen being from primarily, that you showed anyway, from Asteraceae plants, which is late summer blooming. And I wondered if this has any contribution to the low transmission. And does Cryptidia, is it there in the early part of the season, early summer? And what about, I mean, I don't know I don't know how long this particular bee's life cycle is, but I know I see bees in rhododendrons in the early part of the year, so.
Which also has nasty stuff in its nectar that can reduce defenses. So bumblebees are annuals. The queens overwinter, And then, so some of the first bees you'll see in the spring are the bumblebee queens, and they're foraging, but they're also looking for nesting places. I always call them like the original single working mother. Like unlike a honeybee queen that can sort of be in the colony and others do the other jobs, a bumblebee queen at the beginning has to do it all. She has to find a nesting site, she has to feed herself, she has to lay her brood, she has to take care of that first batch of brood, and then once they hatch and those workers come out, they'll start foraging and start doing more of the brood care, and at some point when the colony has enough resources, they'll go reproductive, make males and daughter queens that will leave and mate. At the end of the season, everybody dies except those mated daughter queens that overwinter. How that relates to Crithidia is that, so the only one who can, Crithidia doesn't live outside of a host for more than a few hours. So the only ones that are going to carry Crithidia over the winter are going to be infected queens. Infected queens are less likely to live. So I think this sort of this drop in Crithidia. So early in the spring, there may only be not that many queens that have actually made it through the winter and are infected. But then they start having offspring, they share that disease, they start spreading it, foraging it flowers. So if you sort of sample Crithidia over the summer, it kind of goes up and up and up and up and up. It does seem to drop off a little at the end of the season. Who knows? Is that the Asteraceae? Is that something else? And then it kind of plummets over the winter. So Crithidia tends to be a little bit of a curve like this over the season. Because Crithidia does seem to have a pretty big impact on the survival of those queens, I think it's kind of interesting that there's so many late flowering asters and that might be kind of important.
Yes, very.
Yeah. Yes?
I was just wondering if you thought that using microcolonies in your experiment made the bees perform less effectively because they might be stressed due to the presence of their queen.
There are some studies that suggest that microcolonies are a pretty good way to estimate whole colony performance. There are some that have tried to sort of look at the effect of treatments on both microcolonies and whole colonies and got similar results. Yeah, I mean, these are commercial bees that we use for most of this. They're not that bright. They've never foraged or flown until we stick them outside. Like, they've spent generations being bred in a commercial facility, never having to fly. So there's some compromises that one makes for the sake of feasibility, for sure. Anyway, yeah, you're welcome.
I'm curious if there are plans to identify more species that are good at reducing pathogen loads, and yeah, also second question, are there any plans to do like landscape level projects that assess these patterns, or maybe population level patterns in response to?
I'd love to. So to answer the first question, I'd love to. I need somebody to decide that that's worth spending some money on, because like these aren't particularly easy trials to do, and to say like we did it on 15, give us money to do more. I haven't yet figured out who I should ask for that money from, but I would love to assess more plant species. As far as is anybody doing this at the landscape level, not yet. I kind of flirted with the folks who are doing the solar panel, developing pollinator habitat for solar panels, and if I could talk some people into, could you try this mix versus that mix? And then I could go and sample for bees. That would be one nice way to kind of piggyback that onto somebody who's already doing some kind of testing out different pollinator plantings. If they would let me sort of choose, cherry pick some of the species in the mix, I think that would be a nice way to do that. All of this second stuff is part of this larger Ecology and Evolution of Infectious Disease grant that's led through Cornell. So there's people who have done observational sort of pollinator networks, like not just bumblebees, but all bees, who's visiting which plant species, and then collecting both the bees and the flowers, where is infection occurring, and how does that overlay on traits in both the plants and the bees. So this is part of a larger study, but that's a very big observational study. I'm the experimental person in the collaboration, and I try to carve up pieces that I can manage. Yeah. Yes?
Thinking of bee behavior, do you have a notion whether bees self-medicate when they're infected? Do they choose things that are good for them?
Everybody asks me that. Yeah, it's a great question, which is why everybody asks that. We see very limited, I'd say, there's not a lot of people who've looked at that. There's one really cool study by Leif Richardson, who was at University of Vermont, with turtlehead. Clone glabra, which has iridoglycosides in the nectar, pretty high concentrations. And he made little inflorescences on stalks and added sugar water with high or low iridoglycosides, and then would chase bees around as they kind of entered his array of flowers and record what they were doing on an iPad and then catch them before they left the array. I was like, where do you put those?
Do you think that maybe defers the behavior?
I'm sorry?
Do you think maybe that prevents the behavior?
Bumblebees they're so much easier. I don't know, I've worked with like hummingbirds, and hummingbirds like shoo, 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 and if you get too close, they're gone. Bumblebees are just doing their thing. They're really pretty easy. I mean, maybe that's sort of a somewhat flippant answer, but like bumblebees, you can get pretty close and it doesn't seem like they care. And then he looked at the infection levels in those bees. And he found that if the bees were uninfected, they didn't care what they foraged on. But if they were infected, they were more likely to stay in the array if they visited an inflorescence with high iridoglycosides. Like it seemed like they chose that. That's sort of the best evidence there is. The tent study that I did, we spent a lot of time watching bees visit all the plants in those tents. I didn't talk about it because it didn't come out all that exciting, but we had tents with infected bees and tents with uninfected bees. And we tried to look, do the infected bees prefer more of the low transmission plant species? Do they switch between plants more? We really see no evidence of anything. Yeah, I mean, they have to both self-assess and then know which plants are gonna help. It's yeah. They're not very bright. Yeah.
Well, think about what you're saying, looking in the tents. We were just dealing with small tents and talked about how there wasn't really enough nectar load in the control tents and such. So it would make sense that they didn't have enough food to make that choice.
Right. So that's totally appropriate, and that's definitely going to be in the discussion of that paper that I'm writing up right now. Is that it may have just been that, like, if we'd only looked at the first, hour visits in the morning, maybe we would have seen a signal. But because they're stuck in this tent and all they have to feed on is what we gave them, that they're just going to visit everything. Yeah. So it's not the best assessment. Yeah.
So I'm curious, this figure in particular. So that you see a statistical difference between these. I'm curious whether there's any sense of whether these differences are biologically meaningful and whether these differences can be translated to population level.
I mean, that's why I was honestly surprised. These are the species that I drew from for that last tent experiment. And we saw twice as high pathogen loads when infected bees were stuck with these species for 2 weeks than these species. So it does seem like it actually matters, at least over that scale of time.
You think that will translate to population level?
I guess we'll have to do that landscape scale settle and see. But yeah, I was honest. I had my doubts as to whether my little rinky-dink transmission trials over 20 minutes were really going to scale up to anything larger. So I was pretty excited just about this. Sorry.
So I have a submission for your limerick.
Already, see, this is the best part.
It's something I wrote a couple of years ago, and it has to do with herbivores.
Okay.
So it seemed appropriate. And it goes, many meandering moose will lunch on lugubrious spruce. The resulting depression will teach them a lesson. I should never eat spruce, they deduce.
It brings out wonderful things. It really does. I probably just shouldn't try to top that, right?
No, I don't know.
Maybe just leave it right there. Do you want mine too? Or put this you do. I mean, I'm not particularly good at this.
And I don't know that I found yours. I found these that were posted, which were pollinated related, it seemed like, so.
Yeah, can I go back further? How do I get back, like?
I don't know, I just pulled up your.
All right. I have, okay. Okay. There we go. Here's a nice short one for you, okay? Sunflower pollen can help bees galore, but to have best effects, eat now and eat more. Was a study on the effects of timing and dose. It doesn't have to be much, but it's condensing, taking the whole big paper and trying to get it down to this. Yeah.
So you had mentioned that some of these chemicals are probably antimicrobial,
Yes.
that are ending up in the nectar or the pollen.
Yes.
Is there any evidence, so is most of the evidence suggesting that these just sort of, they're intended for microbes that might attack the pollen, or is there some evidence that the chemicals are also influencing pollinator behavior and specialization of pollinators or anything?
So the topic near and dear to my heart is like, why would plants put nasty defensive chemicals in their nectar?
Yeah, I guess that's what I was trying to ask.
Yeah, which I ended up writing as a chapter, a review paper for my PhD thesis that has spawned almost everything I've done since then. Because it's weird, right? Like, why do you go stick nasty defensive chemicals in your nectar, which is your reward? And so people have done all this armchair hypothesizing, like, oh, maybe it encourages specialization, maybe it keeps nectar from rotting, maybe it protects against nectar robbers and thieves, maybe it makes pollinators drunk and so they don't groom themselves as well, and so then the pollinators, like, all of these people coming up with all these ideas and no data at all. Well, the way I actually got into this was I had a grant with my collaborator Becky Irwin about nectar defensive chemistry and how it might affect plant reproduction, because everybody started with the assumption this must be good for plants because ... And no one had actually shown that these chemicals were good for plants. So we would manipulate nectar chemistry by putting sugar water in flowers with different concentrations and found very little evidence for benefits of plants. But to my knowledge, even that was a while ago, and I can only think of 3.5 studies that have actually asked whether nectar defensive chemicals benefit plants, let alone to start thinking about how the impacts they might have on insects or whether they've been selected to be medicinal as a reward to bees. That feels to me like a bit of a stretch, honestly. Like a pretty, you need a lot of links. Like that assumes that like the chemicals can be detected by the bees and that they'll come back to those plants.
Are the chemicals like uniquely produced in the pollen or nectar, or is it sort of a leaky, like the whole plant is producing these things and then also?
These are all my favorite questions. There are some compounds that seem to be uniquely produced in pollen versus nectar, and there are different compositions of pollen versus nectar. I had particularly thought that nectar, since it's derived from phloem and xylem, that it might be that it's, and if there's any compounds that are transported through the phloem, for example, or xylem, that it may just be hard to keep this stuff out, and it's not very high concentrations in nectar. Pollen seems like a bit of a different story, but I think there's some evidence that what's in the nectar reflects what's in the rest of the plant, but there's also some studies where nectars might have, say, higher concentrations in the rest of the plant or somewhat different compositions. So I think it's both. Is the answer. Oh, yes. Okay.
So does the pollen itself have anti-parasitic effects? Does it need to be metabolite, or does it need to be go through the bee? If you just put it on the parasite, will the parasite die?
You guys have so many good questions. So to put it on the parasite is kind of a tricky thing, but a graduate student of mine who liked much more controlled studies cultured Cryptidia in media in the lab. And he actually did a study doing extracts, like methanol extracts of pollen sunflower and a bunch of other ones, and grew the Cryptidia in the extracts. Cryptidia did great in all those extracts, which suggests to me that this pollen is doing something in the bee, maybe uploading, upregulating immune responses. We're also, I should have shown a picture. Sunflower pollen is really spiky. Asteraceae pollen has a lot of spikes, and we are finally have the funding now to properly play with whether it's really more of a mechanical thing than a chemical thing in this particular case. Yeah. Well, thank you very much.
All right, so we'll probably wind down.